Welcome to Redefining Medicine, an intimate and personalized program that illustrates a different side of the practice of medicine. Our in-depth conversations will focus on the physicians and practitioners who are redefining medicine through their integrative, functional, and holistic approach to health and well-being.
We are thrilled to welcome Dr. Robyn Klein, Associate Dean for Graduate Education for the Division of Biology and Biomedical Sciences at Washington University in St. Louis, and the founding director of the university's Center for Neuroimmunology and Neuroinfectious Diseases. Thank you for taking the time to join us.
Thank you so much for having me. It's really a pleasure to be here.
So you are a physician scientist recognized internationally for your work on the brain's immune system. So let's start there. What is the brain's immune system?
So that's a really interesting question. And so I think I'll begin by giving a little bit of history of, the brain's immune system. So when I was in medical school, we were told the brain didn't have an immune system and that T cells were essentially unwelcome in the brain. But I was also training during the period of the HIV epidemic. And what I was observing, firsthand in the hospitals was that many patients with AIDS, when their immune system and their T cells were essentially obliterated, were presenting with opportunistic infections, and 70% of those were in the central nervous system. And so it occurred to me that perhaps what I was learning was incorrect, and that in fact, not only are T cells welcome in surveying the brain, but that they're neuroprotective and that they're really essential for brain function. And in those days, we didn't know a lot about what we know now. So now, fast forward, what we've learned is that the brain isn't really, as immune privileged as previously thought, but in fact, it does have some more stringent regulation of immune function. And that is really important because we have this sort of hard skull and we really don't have room for expansion of T cells. And so a number of specializations have evolved in the brain to regulate stringently the entry of immune cells and inflammatory responses. And so we also know now that the meninges as a compartment which surrounds the brain has lymphatics, which we also, had been previously discovered, but now we know a little bit more of the molecular interactions that occur and the cellular interactions that occur in that compartment. And we also know that T cells can and do enter the central nervous system, enter the brain parenchyma, that they gather in perivascular components, that they actually cross from the blood to perivascular locations in the meninges and actually climb down, and that there are numerous specializations that allow them to, there's sort of, checks to determine whether they're permitted to enter. And so they have to encounter their antigen, they have to be locally restimulated, and then they can migrate into the CNS, and that this is incredibly important for clearing viruses, that cell-mediated immunity is really the way infections are cleared in the CNS, not just viruses, but also bacteria and parasites. And so we now have a breadth of information about how the blood-brain barrier functions. It's no longer really considered like a wall, but more of a dynamic set of, doors and processes that allow certain cells and certain molecules across, and then allow them to encounter antigen presenting cells which reside in the brain to re-stimulate T cells.
So how do T cells enter the brain?
So T cells can cross into the get into the CNS, as I mentioned, through the meninges, but they don't actually enter the parenchyma. They're mostly within areas of the brain that are called, Virchow-Robin spaces or perivascular spaces on the venular side. And what we showed was that there was a particular receptor on T cells that is a localization cue, and that the abluminal surface of all of these venules, can express the localizing cue. And so what happens is T cells adhere to the vessel as they climb down through a molecule called CXCR4, And I got interested in that because it's one of the receptors for HIV. And that molecule is really important for allowing the cells to enter the CNS through adherence to the vessel surfaces. And then once it encounters antigen, that molecule is downregulated. And then the cells are free to respond to other localizing cues that may be inside the parenchyma. And so one of the interesting things about West Nile virus is that, in fact, neurons can express localizing cues that bring the T cells to them so that they can be cleared of virus.
I have some close relatives who have, one has Lyme disease, and she's suffered from that for years. Another one is just getting over West Nile, and another guy who is experiencing all sorts of like arthritic type symptoms, and found out that he got tested and has or has had Rocky Mountain spotted fever. And of course, Lyme and Rocky Mountain are tick-borne illnesses, and West Nile, and they're bacteria, and West Nile is mosquito. Do they have different ways that they affect the body?
So pathogenesis is very different in terms of how long it takes them to get into the CNS, for one thing. It can take a while for Lyme disease to get into the CNS. So it starts by an infection in the skin, and many patients can have that bullet target hole, target rash around this where the tick was embedded. And so there's some time for treatment before you can actually get it into the CNS. And it's unclear exactly how it gets into the CNS, but it is likely to get in through cells that are infected with it that migrate there. West Nile also infects the skin and can infect dendritic cells, but then it actually goes into the lymph tissues and generates, sort of a low level and short viremia. So there's actual virus particles in the blood. So free virions in the blood. There's also immune cells in the blood that are infected. And so how it crosses into the brain is actually twofold. For West Nile, it can actually cross into the brain first through retrograde transport axonally from the spinal, from the skin to the spinal cord. And in many cases, you can see flaccid paralysis without encephalitis. And we think that's how that occurs because it will kill the motor neurons. But it can also cross the blood-brain barrier as a free virion, and in some cases within immune cells that cross. Mm-hmm. And this happens fairly rapidly. I mean, within a few weeks. If you develop a flu-like syndrome and it goes on to infect the brain.
So we're in the middle of the COVID pandemic, and indeed it's spiking in many areas in the country. And we understand, I think, or have heard a lot about how COVID, affects our respiratory system, but what are its effect on our, cognitive function?
So that's just beginning to be, tabulated and accumulated in terms of cases. But it appears that people who survive it can develop what I said before was like a brain fog, but it's really, a difficulty in concentrating, a difficulty in remembering. And it's unclear how it does this. It also can cause, it seems to be associated with, we don't know if it's causal so much, but it's also associated with other neurologic syndromes. There's been acute necrotizing encephalopathy with parts of the brain basically melting away. There have been ischemic events, strokes associated with patients who present with COVID-19. And some of these other, but there's also been an encephalitis, not a necrotizing, but a regular encephalitis. So there have been post-COVID clinics that have been, sort of cropping up all over the place to deal with any of the post-viral neurologic sequela that patients are presenting with now.
But we're just starting to understand its long-term effects at this point.
Yeah, so there's been lots of papers. There's a couple of good animal models now for this virus so that we can really study its pathogenesis. It's very difficult to study in humans. We don't usually biopsy people who have survived a virus and are otherwise okay, especially because the areas that might be affected when it comes to memory are very deep. And so that would not be an appropriate way to treat a patient because of its morbidity. So, but I think that we'll have some answers fairly soon. And I guess the big question is whether the virus can directly enter or whether it's sort of triggering an inflammatory response because of the high peripheral levels of cytokines and the extensive inflammation that we're seeing.
Right. Yeah. You presented recently at AFREM on their module 3. Can you tell us a little bit about what you spoke about?
So I talked about 2 flaviviruses, West Nile virus and Zika virus, and how studies in our lab using mouse models, and in the case of West Nile, also human tissues, that we, a few years back, uncovered a mechanism by which recovery from West Nile can in fact lead to deficits in spatial learning. Patients who recover from West Nile, about half of them can represent with memory disorders, cognitive dysfunction. And so as of now, there are tens of thousands of people who are living with this. And the most recent epidemiological studies demonstrate that these impairments don't go away over time. They actually worsen and can lead to progressive dementia. And so this raises the notion that dementing illnesses, and we don't know which kind or what fraction or whether it's a subtype, can be caused by viral infections. And in fact, even folks that don't present with neuroinvasive West Nile virus, but develop a flu-like illness, can also present with this. So there's a possibility that small amounts of the virus enter the brain and are cleared, and that that's enough to trigger this process. And so what we showed was that the virus targets all over the brain. It infects neurons, almost all regions of the brain. It doesn't have a specific area it targets. And post-infection, during recovery, microglia remain activated. And in fact, T cells, which is how we started this conversation, T cells are found in high numbers in the hippocampus and cortex. And in fact, we demonstrated that the cytokine secretion by these T cells can trigger the microglial activation. And that also the initial influx of monocytes can dampen neurogenesis within the hippocampus, which is important for spatial learning. So we picked spatial learning because it's been very well established in mice. It's hard to do other memory studies, that you do with humans, such as asking them to remember words and things like that. But we've gone on to show that there's a number of cytokine receptors that are expressed by neural cells that impact on sort of neural correlates of learning, such as generating new neurons in the hippocampus, maintaining synapses and strengthening them through the circuit between the cortex and the hippocampus. And that loss of these functions appears to lead to loss of the ability to learn.
So tell us a little bit more about your work at the university.
So I'm the Associate Dean for Graduate Education, and so I'm the Dean of the largest graduate school at Washington University, in fact, with over 650 students and over 500 faculty that are affiliated. We have now 13 biology and biomedical graduate programs in which students matriculate to obtain a Ph.D. in a variety of subjects, variety of fields, I should say, not subjects, but whole fields. And so I'm in charge of the financial, the student and alumni affairs, and the recruitment of all these students. And of course, I've got directors in each of those realms, but I sort of take it very seriously because I feel like I need to be an advocate for all the students. And I especially am very interested in gender and race harassment and discrimination, and in combating that, in academia. And we have a number of programs now to improve diversity, equity, and inclusion, and we'll continue to do so. There's a lot of work to be done.
As we wrap up here, could you tell us what area of research and study or you're most excited about?
So through the years, my lab has done a lot of work to look at how T cells gain access into the brain. We've looked at how viruses can cross the blood-brain barrier and enter neurons. And we've looked at essentially how the brain protects itself from viruses and clears them. And now more recently, we've moved on to show that the T cells may in fact have pathological effects. And so I'm really excited about understanding how that functions, because most people have resident T cells that remain in the brain near the end of their life. If you look at human brains, you can always find T cells. Mostly they're located around vessels, and in some cases they can be, in the brain parenchyma itself. But the question is, which T cells are protective and which T cells are causing these types of diseases, and how do they remain activated? And is there a way to target them so that you can prevent dementia in patients that have had viral infections? And so that to me is really exciting, and it kind of has brought me back, to my first interest, which was T cells in the brain. And then in my graduate work, I was an astrocyte biologist. And it turns out that astrocytes are, being made instead of neurons in these brains. And so I've come full circle to that again, to look at the role of astrocytes in maintaining cognition.
It sounds like fascinating work. And thank you so much for taking the time to join us today. We really appreciate it.
That was a lot of fun. Thanks so much for having me. It's always great to talk about this.